Have you ever received a beautiful plant in tissue culture, looked at the jar and thought, perfect, clean, healthy, nothing wrong here? And then a few weeks later, after moving it to fresh medium, the whole thing suddenly turns cloudy, slimy, or starts to collapse? This video is about one of the most frustrating hidden problems in plant tissue culture: latent endophytes, bacteria that can live quietly inside the plant without showing themselves at first. And this matters a lot, not only for big commercial labs, but also for small labs, rare plant collectors, and home tissue culture growers. The illusion of sterility. The most annoying part is that the culture can look perfectly clean. The plant arrives in gel. The leaves look green. The medium is clear. No fungus, no obvious bacteria, no smell, no warning signs. But then you remove PPM, or you move the plant to your own medium, and suddenly contamination appears. It feels like something went wrong during your transfer. But very often, the contamination was already there. It was not created by your lab. It was simply being hidden. What is actually happening? Surface sterilization only cleans the outside. Bleach, alcohol, rinsing, all of these are mainly fighting what sits on the surface of the plant. But some bacteria are not on the surface. They can live inside the tissue, in tiny spaces between cells, in old basal tissue, inside vascular tissue, or around old nodes and callus. That is why the old base of the plant is often the most suspicious part. It has had the longest contact with the old medium, the original lab environment, and the internal bacterial population, so a plant can look clean outside, but still carry a hidden problem inside. PPM is not magic. PPM is useful. It can save cultures. It can suppress bacteria and fungi. It can make transport and quarantine much safer. But we should not treat it like magic. Sometimes PPM is not curing the plant. It is only keeping the bacteria quiet. Think of it like turning the volume down on a radio. The sound becomes very low, almost silent, but the radio is still on. When PPM is removed, the bacteria may start growing again. So if a plant only stays clean while PPM is present, that plant is not proven clean, not one-time microbe. It is also important not to imagine this as one mysterious Thai bacteria. Imported tissue culture plants can carry many different hidden contaminants. Some may come from water, substrate, production systems, handling, old stock plants, or the internal microbiome of the plant itself. In different studies, researchers have found many bacterial groups in ornamental tissue culture, including Bacillus, Pseudomonas, Pantoa, Microbacterium, Curtibacterium, and others. So the real problem is not one villain. It is a whole hidden ecosystem, and every plant line may behave differently. The treatment toolbox. There are treatments, but none of them are perfect. Antibiotics can help in some cases, but they should not be used blindly. The wrong antibiotic can damage the plant, fail to remove the bacteria, or simply hide the problem for a while. Copper sulfate has shown interesting results in philodendron research, but copper can also be toxic, especially for delicate plants like small monstera, alocasia, or variegated clones. Vacuum infiltration can push treatment deeper into young buds, but it needs proper equipment and careful control. So the goal is not, what chemical can I add forever? The better question is, how do I recover a truly clean plant line? 3 common mistakes. There are 3 traps small labs should avoid. The first is permanent PPM dependency. If a culture must always stay on PPM to look clean, it is not a clean culture. The second is blind antibiotic dosing. Adding human or broad-spectrum antibiotics without knowing the bacteria can create more problems than it solves. And the third is using harsh toxic chemicals as routine treatment. Some extreme sterilants appear in old literature, but they are dangerous, difficult to dispose of, and not suitable for normal home or small commercial labs. Clean culture is not about using the strongest chemical. It is about using the right strategy. Sort your collection like a lab. A very practical way to think about this is to divide your tissue culture stock into 3 groups. First, quarantine stock. These are new imports. They may look clean, but they are not proven yet. Second, suppressed stock. These are plants that look fine on PPM, but crash when PPM is removed. These are active risk lines. Third, clean index stock. These are cultures that have passed several transfers without PPM and have been checked more carefully. This kind of separation protects your whole plant library, because one hidden contaminated line can quietly waste months of work. The rescue strategy. The most important idea is this. Sometimes you do not cure the old plant. You escape from it. The old base, old nodes, and old callus may be heavily contaminated internally. Trying to disinfect all of that can kill the plant before it kills the bacteria. So the better approach is often Meristem or young bud rescue. You take the youngest, cleanest-looking growing point, the top, the fresh axillary bud, the smallest piece that can still survive, and use it to start a new line. You are not trying to wash the whole history of the plant. You are trying to carry the genetics forward into a cleaner future. Clear gel is not proof. A clear jar is comforting, but it is not final proof. Plant tissue culture medium is designed to grow plants. It is not always the best medium to reveal slow, hidden bacteria quickly. That is why bacterial indexing is so useful. You can take small pieces of suspicious old tissue or liquid from around the base and plate them onto proper bacterial medium. If bacteria grow there, then the plant line was never truly clean, even if the tissue culture jar looked perfect. For more serious work, colonies can be identified with 16S sequencing, so you know what you are actually dealing with. A simple pipeline for small labs. For a small lab, the practical system can be simple. New imports go into quarantine. A few test pieces are moved onto PPM-free medium. If contamination appears, do not panic, and do not try to save every piece. Instead, take the youngest growing point, rescue it, and move it through clean passages. Then remove PPM again and test the line. Finally, use bacterial indexing where possible. This is how a hobby lab starts behaving like a serious micropropagation lab. Not because it is fancy, but because it protects time, money, rare plants, and customer trust. Closing message. The big lesson is this. True plant health is not suppression. True plant health is freedom from dependency. PPM, antibiotics, copper, and other tools can be useful, but they should not become a permanent crutch. For rare imported plants, the future is not in keeping suspicious cultures alive forever with chemicals. The future is in clean stock, careful quarantine, Meristem rescue, and proof. Because every clean culture is more than just one jar. It is a safe starting point for future plants, future sales, future collections, and future experiments. And in tissue culture, that is the real treasure. Not just growing plants, but growing plants you can trust.